So we're going to switch gears here a little bit and talk about biorepositories and our experience with Gulf War veterans. But when I was asked to talk about this, the person that I thought of that should come and talk is Dr. Nancy Klimas, who's got more experience, 30-plus years experience in running biorepositories. And so I'm going to have her start our talk, and then I'll follow afterwards.
What you should have said is that together we have partnered to create biorepositories, and we play two different roles, and we change hats back and forth a lot. It's kind of hard. You got to watch the hands. Anyway, thank you very much for inviting us. First, there's no financial conflicts with either one of us, official statement, and it's always a real honor to come to the National Academy of Science and get to talk. It always feels extra special, so thank you for that. And here's it go. So I'm just going to do a few introductory slides about biorepositories in general. Kim's going to talk about the success of the Gulf War illness biorepository as a good example of why they're important. I know that your job is to make recommendations at the end of the day, and that you don't need to know how to do a biorepository, but rather you might like to frame in your mind's eye as you're listening, what would you like to see in your recommendation, in the type of biorepository you might recommend, the access to that biorepository, and how it might truly help the field as it goes forward. I'd also ask, because I know this is a lung-focused meeting, and you know this, but I'm talking to the choir, but for this on the record, the things that go in the lung get into the body, and there's a whole lot more effects than what's in the lung. So a biorepository has to reflect the toxins that were inhaled and where they went and what they did, and so the types of samples you collect need to pay attention to the broader thing. So that's sort of like a little introduction. So a biorepository, as you know, shouldn't be called a biorepository. It's the wrong name, because a biorepository suggests that you're banking something, like you're storing it, and it's not like money. If you keep it, it doesn't grow and get better. It's really a bad idea. The idea is to get rid of it, to bring it in, have well-characterized samples, and get them out the door to promote science. It's that last step that is always the step that people trip over. A really good biorepository is not a full and complete biorepository. A really good biorepository is one that was full and you emptied it. And you gave the samples to people who are doing innovative and exciting things. I mean, there's got to be some criteria, and you have to be respectful to the donors, that you didn't throw their wonderful donations away. But in the end, the judge of what you did at the end of the day is that you promoted science with that biorepository, and you didn't inhibit the ability for people to enter the field and do their science. The bars matter a lot. So biorepository can be a lot of different things. It can be data, it can be samples of all different types. There has to be a consistent way things are handled. The standard operating procedures really matter in a biorepository. You need to be able to promise the person who gets the sample back from you that you know how it was handled from the moment it was gathered to the moment it got in the freezer, and that the freezer conditions were good the whole long way. So there has to be a really good track record, a template for how you manage and follow those samples. The types of samples can be anything under the sun. We typically have a lot of blood in a biorepository, but obviously when this group better than others, in fact the talk you just heard, a brilliant talk about why tissues matter and why the associated clinical procedural data matters. And so you need to be able to link to the samples in the freezer, the right clinical information as much as you possibly can to what's in the freezer. And then you have to think about what you want in the freezer and what it might be used for. There's a little bit of sand under your feet in science. There's always the next best, better test coming down the line, and you wished you'd held something for it, you know. Whole genome analyses, you can afford that now, how cool. But you can't, you know, again, here's a good example. I don't put up red cells. Who would have thought to put up red cells? Well, it turns out you can do a lot of good stuff with a red cell clot that I had no idea. Someone asked me, and I've been throwing it away. Why didn't I put up, the red cell clot? So there's also a question of what else should there be in that freezer, and what new assays coming down the pike might use those types of samples. What is the linked data? And then something that Kim is really good at, making people play well together. If someone uses your biorepository to create new knowledge, and this is an NIH obligation now, you should be able to then share the information with someone who pulled the same sample and learned something else about it, so that you get the synergistic effect of growing knowledge around that. And so you have to create a structure for that sharing and a set of rules that people agree to play by when you create that. So a nice recommendation when you're making your recommendations is to think this all the way through. The other thing is this cool concept, which Kim included in her new Biorepository grant, which is this idea that there could be a virtual biorepository. That would be linking everyone who has samples in some rule book where they play nicely together and share their samples. So I have a lot of samples in my freezer that had nothing to do with the biorepository. I'm happy to share them. I might have things in my freezer that weren't a part of somebody else's plan, or someone has tissue. Spinal fluid. Oh, it's a wonderful resource, but who's got spinal fluid? So the idea of linking all these kinds of things together and finding who's holding what in their freezer, doing surveys to figure out who's got what, and will they play nicely together as a virtual group to hold a virtual biorepository. Same thing goes with the clinical data that goes hand in hand with that. And so that's kind of a new idea, but it's very doable, and it makes international collaborations more possible. So the big issues, respecting donor privacy, being very careful about privacy and knowing what the samples were consented for. That has to be a part of the biorepository, is the actual informed consent the patient signed. Has to be linked to that set of samples in a biorepository, so you know when you have to throw them away because they've been consented for so many years, and what you can do with them. Some of them are not consented for, say, genomic studies, and you have to know. So there has to be a way in your platform that you know what you're allowed to do under the restrictions that were placed in the original assessment. Something that is very important, you heard it in the 585 talk conversation, was encouraging people with biorepositories to consent people to be reconsented for future studies, to be allowed to go back and do that double tap. Because then you don't have to throw it away. You can go back and say, hey, you told me to throw the sample away in 20 years. I'd like to still use that sample. Do we have permission to continue and get additional IRB coverage? So being a responsible custodian across the board. Later in this slide set, you'll see when Kim's talking that the National Heart, Lung, Blood Biorepository Guidelines link is in this talk, and you have the talk. So that would be a very good guideline book for recommending people look at that if they are asked to create a biorepository. I think that's my last slide, and it's yours. Thank you.
So thank you for that overview, Nancy. So now I'm going to give some examples from some of the work we've been doing with biorepositories. So I primarily study Gulf War illness. So this is 1991 Gulf War veterans that I study. And what we did was we started a Gulf War illness consortium study, which was a large study. So we have 16 collaborators, 9 study sites around the country, and we actually have an international collaborator as well. And it was designed to bring preclinical and clinical researchers together to speed up the process of determining biomarkers for Gulf War illness and developing targeted treatments. One of the things we also did was we created what we thought was a small biorepository. And so we had some money to do pilot studies with the consortium, and we thought, well, we'll use it for that. We'll share it with some collaborators. And it turned out that it was a much bigger thing than we had ever anticipated. Maybe Nancy anticipated it because she initially suggested we think about putting a biorepository in. But I liken it to the movie The Field of Dreams. If you build it, they will come, and boy, did they come. So we heard from many, many, many Gulf War researchers interested in using these samples. What it became was an opportunity to have access to these well-characterized patient samples, and the number of bench scientists who responded were incredible. So we had a lot of scientists who on the preclinical side, maybe they do an animal or cell work, and they wanted to see if it translated to the clinical side with clinical samples. And we were able to provide that. So we provided a lot of pilot study data from sharing this biorepository. And the GWIC experience was really positive for that reason. And it made us think, wow, there's a real need here that's just not being met at all. And so this was something we thought, we could really do in this small way, but then in a bigger way with our new study called the B-Brain, the Boston Biorepository and Integrative Network for Gulf War Illness. And just from our GWIC studies, we've got funded, initially in 2013. And these are all the studies that actually got funded, mostly from DoD, some from VA, some from NIH, from using our samples. So this was just tremendously successful, as you can see. We were able to provide samples to investigators who maybe didn't have the clinical expertise to go out and get these samples, but they had a really good biomarker idea or a really good treatment development idea, and we were able to share samples with all of them. So these were blood studies, epigenetic DNA studies. These were induced pluripotent stem cell lines that we were able to create and share with others. From that, we were studying neuroinflammation, so we were able to develop neurons and astrocytes from that, and then mini brains. But you could use this for lung tissue. That could easily be created, and at Boston University we have a stem cell center that does this on a regular basis, so that's something worth considering. We also could develop lots of different treatment trials based on some of these markers. So this is just sort of a platform of how just sharing of samples can just exponentially increase your funding, but also your ability to move a field forward quickly. You know, with Gulf War veterans, we were in a position where almost 30 years in, we didn't have good biomarkers. We didn't have a real handle on the mechanisms. We had to bring all the experts together to really work quickly to do this. And I think we're making tremendous progress in this amount of time that could be a model for other fields, including this one. And so our key research accomplishments, we started 10 studies in our consortium. We had 7 additional pilot studies funded from this. We're working on now multiple different treatment trials based on some of these biomarker outcomes. We collaborated with 19 additional biomarker studies, as I showed, multiple publications and symposia from this. And I think the most important thing is we brought in new investigators into the field by sharing our samples. And so these new investigators that we brought in were not only perhaps postdocs working in the field of some of the people that we were working with who could then have their own samples to start their own studies, but also really knowledgeable people working in fields outside of Gulf War illness that we were able to bring in because we were able to share samples with them and entice them to come into the field. And we really were able to fill gaps in knowledge that we didn't have by bringing these investigators in. And so our new study, as I mentioned, is the B-Brain study. So this is a much larger study now where we're going to collect samples prospectively from multiple different study sites and also retrospectively, as Nancy mentioned. So we actually said, well, there are a lot of studies out there who have stored samples that are really important that, you know, it's hard to replicate. So there are folks who have stored CSF, which is difficult to obtain, and we don't want to be sticking our veterans multiple times. If we can share this and make a virtual repository. So that's what we're doing with our B-Brain. As one of our aims is to build this virtual repository where, you know, maybe someone has serial bloods that they took from a participant, you know, or from a veteran 30 days in a row. Not a lot of studies are going to be able to do that. And so we've actually gotten many studies to sign on to share their samples to add to our repository. And in our repository, we are going to have multiple different kinds of samples, so everything from blood, CSF, DNA, stool samples for microbiome work, all different types of samples. We also will have clinical data to go along with that. So I'm trained as a behavioral neuroscientist, so it's important to me that we have these behavioral pieces to go along. So we have cognitive data, we have validated symptom questionnaires, we have other measures of brain imaging and Fitbit measures of sleep metrics, et cetera. But it's really important that you have your data to go along with your samples for your behavioral outcomes. And it's really important that a field uses the same metrics to do this, because if you don't, you're comparing apples to oranges versus apples to apples, right? In the B-Brain, we're going to have 500 Gulf War veteran participants, and we will do this at 4 different study sites. So it'll be in Boston, Miami, New York, and San Francisco. And these samples will be available for sharing. Here's just sort of an overview of what happens in our repository. So we will get the samples from our participants, and of course aliquot them down to plasma, PBMCs, serum, et cetera. We'll also get saliva, urine, and stool. An important thing I just want to note here is that you can think about what did I do? You can think about a repository in a couple different ways. So you can aliquot in large ways, so 1 ml, 2 ml, or you can keep hitting this. Whoops, what did I do here? Oh, sorry, everybody. You can aliquot down into larger samples or smaller. Not a lot of studies do both, because it takes a lot of technician time to do that. But what we found was if you do it both, so if you do 1 mLs and then you can actually do it down to 0.25 mLs, you can share with a lot more studies. And so what we found was that was a way that we really could actually share with 20 somewhat studies, and we have more to share with others because we've done that. So I just want to make a pitch for really thinking about if you're going to do a repository, how are you going to aliquot down? And then, of course, I learned from Nancy that always ask, what's the minimal amount you need in your samples? Because everybody typically will tell you twice as much as what they actually need. Oh, did I do it again? Oh, my gosh. Okay, so what are the current military biorepositories? So I told you about the ones that we're running. So we have the GWIC and the B-Brain, and also the virtual repository that we will have as part of B-Brain from other people's stored samples. I'm sure there are other stored samples that could be used for burn pit exposures as well. The additional military repositories are the VA Biorepository Brain Bank. It was created in response to ALS in Gulf War veterans that was increased, and so they actually got brain samples from them. It then expanded to Gulf War illness, to PTSD, and to TBI. And this group is really set up to get autopsy samples. They're set up across the country. They do this for brains right now. They could do this for lung and other tissues, so just a consideration here. They're really well ready to go and funded to do this kind of thing. There's the VA Million Veterans Program, and this has survey information and some limited exposure data. I think there is a burn pit question there. I think they have 800,000 participants enrolled to date, which is really tremendous. And I believe that you have to have merit review funding from VA or a special RFAs to be able to access their samples. There's a CSP 585 that you heard about today, the VA study, the Gulf War era and Biorepository. They got 1,200 veterans to enroll of their original 10,000 they were trying to get. They did get 10 mLs of blood from the participants. They have DNA and survey data available for sharing, and they do have an established method for requesting access to the samples. They are sharing DNA and survey data, and I am a recipient of their survey data, so I know that that is available. We're using that for a women's study that we're currently running. But I believe the plasma samples are actually on hold for sharing right now because they only got 10 mLs of the blood, and they really are trying to wait to see what the most important studies are. So again, a pitch to get more sample rather than less. I should mention in our biorepository we are collecting 67 mLs of blood from our participants, and we haven't had any problems. Initially I thought, oh my gosh, they're not going to do this, but there was not an issue at all. They've been very happy to give that blood because they know it's going to be shared for other studies. So if you are thinking about building a repository, think bigger. Get more sample to be able to share. The other studies that have samples are the DoD Serum Repository, I think you heard about a little bit today, and there's 60 million samples there. That's a tremendous amount, but you have to have a DoD principal investigator for use of those samples. And of course, the NHLBI Biorepository is the gold standard, but they don't have military specimens that I'm aware of. But this could be a fantastic comparison group. So what are the challenges of using the military, some of the repositories? So I did mention a couple. So some of them have collected a small amount of sample, and then they hold on to it with the idea of we want to wait for that perfect study, but then sometimes it never gets shared. So that's something that's an issue. Some of them require extensive and lengthy approval processes that can go up to multiple chains of approvals and can delay studies tremendously. So what we found was that a lot of people wanted to do pilot studies, came to us in our consortium because we were able to get them samples much faster. So if there's a way to speed that up, that would be an important thing. They often require a principal investigator to be at VA or DoD to get these samples, which isn't a problem if you are there, but if you're not and you're an outside investigator, that can be a challenge. And so we tried to design our new repository with these things in mind to try and be able to share widely with government and outside of government investigators, because we really want to bring the experts in all fields together. What are some exposures that the burn pit and airborne hazards veterans might have had that might be involved in your thinking of what types of biorepository samples you might have. So there are particulate matter and respirable sand that we know were big exposures, heavy metals and dioxins, and of course we know that those will stay in tissues for quite some time. So if you're thinking about tissues like fat tissues and things like that, those are things that you might want to put in a repository. Diesel fuels, chemical and paint products, medical and human waste were some of these, solvents, plastics and rubbers. We heard a little bit about dioxins this morning, and we know that they are carcinogenic, and so I know we're talking a lot about lungs, but we might want to be thinking about different types of cancer that may be happening down the line, so getting the right things in a repository will also be important. Okay, and how do we better understand chronic health risk pathogenesis? So one way is to make sure that your data of the exposures are connected with your biomarker specimens. That's going to be really critical. So you want to determine, for example, the frequency of the exposures as part of a health risk assessment, for example. So you could ask how many days were you exposed, even how many hours a day, if it's part of your occupational specialty. Then you can do a health risk assessment based on this. You can create common data elements for your exposure assessment so that everybody's using the same tools to do exposure assessment. If you're not doing that and you're doing studies of different specimens, you really don't know if you're comparing apples to apples or apples to oranges. So I would highly recommend, at least there's some kind of a common data element to be used. We've done that in the Gulf War illness field, and it's been quite successful for us. And you can also consider things like air modeling, so GIS modeling, and I was very encouraged to hear that some groups are already thinking about that this morning, and I think that's going to be really critical and very important. The other things you want to think about is considering detoxification systems and compartments at risks. So things like liver or kidney might be important here if we were talking about exposures. And you would think about when you're designing a biorepository for health consequences that might be seen years after the exposure rather than 6 or 9 months. We're talking about, health effects many years later, so that's going to be important when you're designing any kind of a biorepository. And so with that in mind, what could you add to a biorepository? Whether it's a virtual one of things that already exist or a new one, you might want to be thinking about things like blood and cells, sputum and saliva. If you know veterans are getting biopsies, you might want to think about trying to get that lung tissue or their kidney tissue. Urine and stool also can continually detoxify or pick up detoxified materials. So these are things you might want to consider. And happy to take any questions.
Questions from the committee, Frank?
Can you tell us a little bit about, in your biobank, what kind of phenotypic information you actually have on the people that are in the biobank?
I'm sorry, can you
Can you tell us something about the kind of phenotypic information you have for those people that are in your brain bank?
So
B brain bank.
In the B brain, yes. So we're going to have. So this is a study based on Gulf War illness. So we're characterizing them whether they have Gulf War illness or not, and that's by self-report.
How have you collected the information that relates to those people?
So this is self-report of their symptoms, is how we're characterizing them as Gulf War illness cases or not.
But he's asking about the phenotype, so the neurocognitive. He wants to know the workup.
Oh, sure. So yeah. So we're doing. I think I had a slide there that was the full overview of what we're doing. So we're getting blood samples. We're getting urine. We're getting stool.
I'm more interested in the phenotypic clinical side of the information you're getting about each person's specimen.
Yes, so we'll have cognitive outcomes. We have validated survey markers of symptoms, pain, fatigue. We have questions about respiratory problems, about, although that's not our focus, so we don't have a lot there, about pain, cognition. We have brain imaging that will be correlated with that as well. And we have measures of heart rate, sleep, and exercise.
Other questions? Sorry.
Yeah, just a quick one on the prospective analysis. I mean, one of the issues for us is actually how to make use of repository data when there are challenges of what do you compare with what? How do you move it beyond a pure sort of descriptive type of analysis? And obviously you have some measures of exposure and that sort of thing. But
Yes.
In the prospective studies, are they actually just longitudinal, or do they cover periods of deployment, pre and post, that sort of thing, or?
Good question. So they are during deployment. So we are asking them about their Gulf War deployment. So 1991 Gulf War deployment is what we're looking at. And the longitudinal piece is we've seen them originally for our Gulf War Illness Consortium, and now we're going to see them for the B-Brain. It's 3 to 5 years later. So it's not pre-deployment.
They're years.
It's a year span of post-deployment.
Yeah.
Right.
It's all post-deployment.
Yeah, we only have.
It would have been wonderful if we had pre-deployment, but we did not.
I really appreciate the culture of sharing and, pushing science and that kind of thing. A couple questions. One is, how do you think about authorship? And then the second is, thinking about things like urine collections. That can be useful depending on what you're doing, but if you're, for example, wanting to understand metal exposures and you use urine as a biomarker, if you don't acid wash the collection
True.
device, then you can't do it. And I just wonder if you could help us kind of think about lessons learned along the way around some of the kind of linking the exposures to the samples that are actually in the biobank.
I like the question because today the CDRMP applications are due, which is why you may see me texting, sorry. But we have, 15 people that are submitting today
We do.
using our biorepository?
Yep.
And so these very questions, authorship, we had one that wanted 5 cc's of blood. Well, we couldn't provide him 5 cc's and have anything left for everybody else. So we asked that he include in his application enough money for us to redraw on this longitudinal cohort sufficient sample, an extra of assessment day. If someone came in and they wanted a chelated urine sample or something like that, they could ask for that. We'd have to work it into the design. We'd get a second informed consent, and they could come. We would work with them. We would create within the B-Brain a
And
way
we've-
to collect the sample they want. A little hard to predict what everybody needs, but if they come along with a need that we didn't think of, we try to work with them to
And we've
to
done
solve
that. So we've built in callback studies for folks that we couldn't readily share what they want, but we have a cohort that we can do that very easily. And we actually did consult with some folks about things like urine and heavy metal, for example. We did do that and said, what's the best way for us to do this? How should we collect this? How should we then store it? So actually, that's a great question because we've actually been spending a lot of time talking with folks at Dartmouth University. That's their expertise. So great question. Thank you.
So a comment and a question. Nancy, that was a great statement that a good biorepository is an empty biorepository. They actually usually run contrary to that. Everybody tries to save
Yeah.
the materials for
for the rainy day
for
that never
golden,
comes.
that golden opportunity.
Yeah.
One of the things you didn't mention, the DoD biorepository has the advantage, however, of collecting samples during periods of exposure. And as I understand it, the way you've set up the biorepository, it's typically some point after the exposure. And I wondered, do you think there's a limit or an endpoint after which you'd no longer want to collect the sample? I mean, is it a year, 5 years? Is there, as you've set this up, any limitation to it?
I think it really depends on the question you're asking, and that's why I talked a little bit about that question about if you're thinking about exposures and how long you can detect an exposure, for example, it depends. So it really depends. So, for example, dioxins may be in your system for 7 to 11 years. So you still are well within the window that you could be thinking about that, for example. So it really depends on your question. Now, for Gulf War veterans, they're still ill, so we're trying to get biomarkers, and there's no reason for us not to be doing this to identify, what's a biomarker of illness versus exposure versus how do we use that to target treatment.
I heard Dr. Naveau give a great lecture at the MECFS conference at the NIH last year, and he was making us be very clear in our thinking that there's a very big difference about the onset of an illness and the persistence of an illness. And that all of us that are doing 30-year later research on these very ill people people pretending like we can somehow bring ourselves back 30 years and really know what the vulnerability and risk and events were then. We're not going to be able to do that in Gulf War illness. It already happened, 1991. Sorry, it's over. We can't do that. But in burn pit exposure, an ongoing problem, you have the advantage in this field of designing a study like Colonel Morris, which where he had post deployment data, where you could think about it. But even then, you have to separate your question between what were the risks and the triggers versus what are the mediators of persistence. Mediators of persistence, you can go right ahead and measure. Please do measure. And the other thing, please don't forget what's going to happen next, because we are in that spot. In Gulf War illness with a neuroimmune oxidative stress brain, we're anticipating Parkinson's disease. It's too soon to see it, but the brain's the right brain for it, right? So we're assuming we're going to see, but we don't know, degenerative, higher risk things. So we're trying to understand an illness in that amazing space where you could actually do something in time to prevent the next step. And how do you convince the funding agencies that that's a good investment? Because it's going to take years to know whether or not you actually achieve that goal. So there's a lot about this. And these people that we're dealing with the burn pit exposures, with their tremendous diversity of exposure, of toxic exposure. I'm an immunologist, so I'm thinking, the vulnerabilities that are going to happen 10, 20, 30 years from now in these folks.
Sorry, any other question? If not, thank you very much.